Hello, my name is Thomas Vinar and I am going to tell you about plASgraph, a new tool for detecting plasmid contigs from an assembly graphs. Plasmids are small molecules that are physically separated from chromosomal DNA. They replicate independently and they are typically circular. They facilitate efficient horizontal gene transfer in bacteria, and they can, for example, carry antimicrobial resistance genes. Therefore, it is a very important task to identify plasmids in genome assemblies. If we are considering a finished genome assembly, the plasmids are typically easily identified since they have a different size and they have a different sequencing coverage, GC content, and specific gene families are typically present. However, it is much more difficult to distinguish plasmids in unfinished genomes, which are, for example, those that are created from short-read assemblies. To demonstrate why this is a difficult task, here is a figure illustrating the structure of a genome assembly from short reads. So each of these vertices in this assembly graph represents one of the assembly contigs. Two vertices are connected if they possibly follow each other in the genome assembly. So each DNA molecule is represented by a wall in this graph, and we have marked these vertices by different colors depending on whether they are chromosomal, whether they belong to a core genome, so this is a blue color, or whether they belong to one of the two plasmids present in this sample, in particular red and green. You can see that the structure of this graph is kind of unwieldy and that it is not a simple task to identify which contigs belong to chromosomes and which belong to plasmids. In order to build our tool, we have introduced several innovations to previous machine learning approaches to this problem. Basically, this is a classification task. The first innovation involves use of a graph neural network layers in our neural network. Basically, the graph neural network layers work as follows. We take a particular vertex and we look at its neighbors. Each of these vertices have a particular feature vector associated with them. These are combined into a single feature vector by a weighted combination. Then classical dense layer and the activation function is applied to obtain a result for node X. The graph layer typically processes all the nodes at the same time and can be applied multiple times in order to propagate information from more distant nodes in the graph. The second innovation addresses a problem where many tools actually need to be trained for a specific species. And this is probably because in a machine learning process, they learn how DNA of a specific species look like, but not learn general features of how plasmids differ from core genomes. In order to do that, we have introduced relative features that mask the features of a specific genomes. For example, instead of a GC content of a contig, we use a difference of a contig GC content from the overall GC content of the sample. And we don't use K-mer distributions. Instead, we only use distance of a particular K-mer distribution of a particular contig from the overall K-mer distribution of sample. And we also only include difference of contig coverage from median genome coverage. We also include several other features that help us to distinguish whether the relative features are estimated reliably or not. And these are in particular contig length and degree of a node in the assembly graph. The third innovation is our treatment of so-called ambiguous contigs. Those are contigs that are shared between both core genomes and plasmids. And on this figure, you can see the overall structure of our neural network. And you can see that we have two different outputs, one corresponding to plasmid score, one corresponding to chromosome score. And these two outputs are inferred from the same information, but they are inferred independently. So it means that we can identify the same contig as both being a plasmid and a chromosome, and this would correspond to an ambiguous contigs. We have compared our tool to two recent tools, ML Plasmids and plasforest. ML Plasmids is a tool that is typically used on the same species that it was trained on, and plasforest is interspecies tool that also uses sequence homology. By comparison, we have demonstrated that we can classify accurately much smaller contigs than both plasforest and ML plasmids, which only typically work with contigs longer than 1000 base pairs. Also, our tool can be successfully applied to novel species, so those that we didn't train on. We have better results than plasforest even without using homology information. And finally, our results are more consistent with assembly graph information. You can see more detailed results on our poster or in the included preprint. Thank you very much for your attention.